Good evening, one and all. My name is Meenakshi Sundaram Palaniyappan. I am serving as the project director of the Center of Excellence for Innovations as well as the head of Department of Biotechnology, Agriculture College and Research Institute, Madurai. Today, I am going to talk on the different types of culture which we encounter in a typical tissue culture laboratory. We all know tissue culture is a applied biology wherein we try to multiply our target plants in a large scale. So, what we do in tissue culture is we try to begin with an explant, which is nothing but a small bit or a piece of a plant which is inoculated onto a artificial medium with the objective of either mass multiplying plants or producing secondary metabolites or use them for storage for a longer time. So, in a typical tissue culture laboratory, different types of cultures are encountered that include Callus culture, cell suspension culture, shoot tip culture, meristem tip culture, anther culture, pollen culture, ovary culture or ovule culture, as well as embryo culture. When you look into the different types of explants, we can now understand that we can use both vegetative tissues as well as reproductive tissues. Vegetative tissues include your shoot tips, meristem tip, leaf bits, stem bits and so on, and your reproductive tissues include your anther and ovaries. So, in this lecture, we are going to discuss 4 different types of cultures, namely Callus culture, cell suspension culture, shoot tip as well as meristem tip culture. To begin with, we will start off with Callus culture. So, before we try to understand what is meant by Callus culture, we should define what is a callus. So, callus is a mass of cells or actively dividing cells with different that are in different levels of differentiation. These calluses are helpful in different aspects. So, the callus induction process includes different stages, namely induction, division and differentiation. So, to explain this, when we take a explant, for example, a leaf bit with the purpose of inducing callus, we try to induce the leaf bits to reprogram themselves to give us mass of cells that are rapidly dividing, for which we need to play with the hormones such as auxins, cytokinins and so on. So, with regards to callus culture, the balance of auxins and cytokinins plays a major role. So, when we place an explant with a balanced level of cytokinins and auxins, which includes both the internal tissue level hormonal concentration as well as the medium level hormonal concentrations, the cells get reprogrammed. They start undergoing division and they start to differentiate. As we all know, differentiation is a process by which rapidly dividing cells acquire special functions. Here in the case of callus induction, the cells, they are not going to acquire any special function. Instead, they are going to lose their function, specialized function, which we call it as de-differentiation. Differentiation includes redifferentiation and de-differentiation. So, callus formation is a process by which differentiated cells, they undergo de-differentiation. So, they become functionless, but still they keep on dividing. So, when we look into the different types of callus that are available, you can group them into based on different criteria. First, based on the texture, you can divide it into hard callus as well as friable callus. Based on the pigmentations or the color of the culture, you have colored calli also. Among these, among the friable and hard calli, friable calli they take up a lot of roles in tissue culture, whereas the hard calli they are discarded. If we are able to obtain a mass of cells or your callus from a single cell, the cells are going to be uniform, or else it is going to be the reverse. So, let us try to understand how a callus develops from a leaf as an explant. So, here in this image, I am trying to represent a longitudinal section of a leaf bit, where when we inoculate leaf bits, the cut ends are exposed to the media. Also, the top surface is exposed to the environment, whereas the bottom surface of the leaf is exposed to the nutrient media. As we are all aware that calluses are mass of outgrowths which develops when a plant is wounded. So, with the objective of the main purpose of callus culture is to induce callus from a differentiated tissue. So, what happens is when we prepare the explants, we are manually damaging the sites. Also, we can use some tools to create wounds on the leaf surfaces. So, your callus they can emerge from the damaged leaf surface or from the palisade or your spongy parenchymatous cells that are seen between the upper and the lower epidermis. So, this callus has to be maintained during tissue culture based on the objectives. So, the callus can be stored for a short period of time or it could be for a long period. When I say short period of time, what I mean here is the callus has to be multiplied in large numbers, so that the desired objectives of either organogenesis or taking up cell suspension can be taken up. When I say cold storage, the callus induced will be stored for a long time, after which whenever a need arises, the callus is obtained and taken up for experimentation. With regards to the short period storage, what we actually do is we try to induce callus. We choose only the friable callus, and the callus which is as a huge lump is broken into smaller chunks. These smaller clumps or your chunks, they are transferred to a fresh medium that was responsible for inducing the callus. The hard callus is discarded and this callus is subcultured once in every 21 days, and this subculturing can be happening for up to 6 to 8 weeks. The problem associated with callus culture is browning, aging and death. So, the callus, not just induction is important, the cells have to be divided in large numbers and they can be partitioned for subsequent experiments. This depends on the objective of the researcher. So, the callus culture, why is it used, or what are the uses of callus culture? So, the callus culture can be used for regenerating a whole plant either by forming the shoots or the roots or roots and shoots parallelly, which we call it as collagenesis, rhizogenesis or colorhizogenesis. The callus cells, they can also be programmed to form somatic embryos which are bipolar structures. The callus cells, they are also used in developing cell suspension cultures which are useful for secondary metabolite production. Callus cells, they are also used for protoplast isolation which are used in protoplast fusion experiments. We all know that genetic engineering is a buzzword during the last 15 or 20 years. Genetic engineering is a process by which we try to introduce genes from a sexually incompatible species into another species. For an example, the introduction of BT genes into rice, brinjal or any other plant for the production of insect tolerant crops. Now, the recent trend is genome editing, wherein we try to bring in some modifications. Be it genetic engineering or genome editing, without Callus, nothing is possible. So, Callus plays a major role in biotechnological research. Now, we move on to the next topic, cell suspension cultures. So, the term cell suspension refers to growing cells in a suspended medium, growing cells in suspension, for which we need to use a liquid medium. On the other hand, Callus culture involves the use of solid media. So, in cell suspension culture, what we try to do is, we try to grow the cells by suspending them in a liquid medium. So, the starting material is always a friable callus. So, large callus clumps are taken, they are broken down into smaller chunks or smaller portions, and they are transferred to a fresh medium. And these cells are ensured to have a minimum cell density. These cells have to remain suspended, so we need a shaking apparatus, for example an orbital shaker. So, to the medium, for enhancing the cell division, we try to add auxins. Also, after the addition of auxins, the cells are put on an orbital shaker, where 50 to 150 RPM is maintained. So, the cells are constantly agitated, so that cells do not aggregate. The cells remain suspended as individual cells, and they remain in suspension. So, the growth of the cells in suspension is analogous to the bacterial growth. We all know that bacteria, when it is cultured in a liquid medium, the growth starts with a log phase, which is followed by a exponential phase and then a stationary phase. Identically, the callus cells are grown until they reach the stationary phase. So, this cell suspension culture is carried out until the stationary phase, after which they have to be subcultured. If we are able to develop suspension cultures from a single cell, it is going to be uniform and is called a single cell suspension. The characteristics of cell suspension cultures include the cells are they are highly separated. You expect a high degree of cell separation. They are of uniform cell morphology. They have distinct nucleus and a dense cytoplasm, which is a mandate for a callus. The tracheary elements are well developed. Tracheary elements are nothing but the water conducting elements that are present in these cells. Each cell takes up about 24 to 72 hours to divide into 2. So, depending on the doubling time, the frequency of subculture is going to vary. We can also witness enhancement of the ploidy level in the cells that are in cell suspension. So, these are the characteristics of a cell suspension culture. Having known the characteristics, it is necessary to understand what are the utilities or advantages of cell suspension culture. The cells under suspension are used primarily for secondary metabolite production. I hope everybody knows what is secondary metabolite. It will be explained further in the subsequent lectures. The cells can also be used for protoplast isolation, which is further used for protoplasm fusion experiments. The cells can also be used for developing somatic embryos. If we are able to develop a protocol for somatic embryogenesis, it is going to be groundbreaking, because the level of mass multiplication in vitro is going to be huge. Standardizing the development of somatic embryos is very critical and it requires a lot of expertise. Now, we move on to the next topic, that is meristem tip culture. I hope everybody knows the difference between a meristem tip as well as a shoot tip. So, the image given here, it represents a shoot tip that contains the apical meristem, a little amount of dividing cells beneath the apical meristem, followed by young leaf primordia. So, we have to understand the clear distinction between a meristem tip as well as a shoot tip. I will tell you the characters of a meristem tip and then we will see the uses of meristem tip culture. So, a meristem tip is nothing but, or a meristem is an area in a plant tissue where the cells are rapidly dividing. The meristems are approximately 0.1 to 0.5 mm long if we collect them from the apical meristems. So, the meristems are characterized by rapidly dividing cells. They are functionless, they just keep dividing and they promote the growth and development of plants upon differentiation. Your meristems can be classified into apical meristem that are seen along the roots as well as the shoot apex. Lateral meristems, they are responsible for the growth or the horizontal growth of the plant tissues, that is the increase in the girth and the width of the plant stems. So, these meristems are seen in the vascular bundle. The last group is intercalary meristems. They are seen along the nodes or your leaf axils. So, we have to understand where these meristems are lying, so that they can be potentially exploited for meristem tip culture. So, the main reason for selecting meristems are in such a way that they are like callus, they are always in an undifferentiated state, they are rapidly dividing. Coming to the advantages or uses of meristem tip culture. Meristem tip culture is used for virus elimination from virus infected plants. Say I have a very good variety which costs thousands of dollars, but it is infected with viruses. If I have to save the plant or mass multiply it without viruses, the only way to do is using meristem tip culture. Why meristem tip culture is used for virus elimination will be explained in the next slide. Meristem tip culture is also used for micropropagation of important plants. They can be used for germplasm storage, which is one other area of biotechnology or your plant tissue culture. Meristems could also be used for genetic transformation experiments. The main disadvantages include you need a skilled expertise because we need to isolate meristems, for which we need stereozoom microscope. So, a person who is skilled in using microscopes and who has a knowledge on the botany of the plants is essential for doing meristem tip culture. How virus free plants are produced? So, this has to be answered. Please remember that viruses are non-living entities. They try to grow, they multiply only in the living organisms. These meristems undergo rapid cell division, as I told you earlier, where the rate of cell division is very high compared to that of the viruses. Also, the meristem tips contain high or rich amount of auxins, which are expected to affect the nucleic acid metabolism of the viruses. Please understand that nucleic acid metabolism is essential for the replication of viruses. Also, The meristematic cells, they are not connected to one another because they lack vascular bundle. Please understand that viruses move from one cell to another through the plasmodesmata or the vascular bundles, which are absent in the meristematic tissues. On the other hand, all the cells in vegetative tissues or the well differentiated tissues are connected by plasmodesmata. So, these are the reasons, some of the reasons why meristems are chosen, meristem tip culture is chosen for producing virus free plants. Also, it is hypothesized that some unknown mechanism for viral inhibition also is existing or prevailing in the meristematic tissues. Next, we move on to the topic of shoot tip culture. Shoot tip culture is nothing but an extension of your meristem culture, where we take explants that are longer in length. So, shoot tip culture includes the meristem tips as well as the young differentiated leaf primordia, as it is given in this image. So, the characteristics of shoot tip include, each shoot tip, you can define a shoot tip to be a part of the plant which is about 0.5 mm to 5 centimeters long. This shoot tip, use of shoot tip for in tissue culture is also called as clonal propagation or micro propagation. The main reason here is we try to exploit the meristematic cells that are present along the axils of the leaf primordia. The different stages that include in shoot tip culture are selection and sterilization of the explants, establishment of axillary buds. Please understand here that this is the shoot tip and you see the leaf primordia. Along the axils of the leaf primordia, you have the axillary meristems. Say for example, in this figure, you have 6 axillary meristems. So, when we try to multiply this from one single shoot tip, we can get minimum of 6 growing shoots. So, that is the power of this shoot tip culture. More the number of leaf primordia, more the number of axils. So, more are the chances of getting multiple shoots. After establishment of these meristems in the axillary buds, multiplication happens in the culture and then we take up rooting of the in vitro shoots. So, shoot induction will be followed by root induction, followed by hardening of your in vitro raised plants. In shoot tip culture, a maximum of 7 times the explants are subcultured, and the main problems faced here are browning of explants, contamination from microorganisms or the phenolics, and somoclonal variation. Larger the number of cycles for subculturing, chances of getting somoclonal variation is very high. So, experts recommend restricting your subculture to maximum of 5 or 6 times in any of the shoot tip culture experiments. Also, vitrification happens. Vitrification is nothing but accumulation of water crystals that is going to kill the plant. In this lecture, we have learnt the basics of Callus culture, cell suspension culture, shoot tip culture, meristem tip culture. So, in the next lecture, we will learn about anther culture as well as pollen culture, ovary and ovule culture as well as embryo culture. Thank you.